Hello, everyone. I'm delighted to be here today. I want to thank the conference organizers for inviting me to share my recent study with you. Before I dive into my presentation, let me give you a quick introduction. I am Reyhaneh Abbasi, a PhD student at the Acoustics Research Institute in Vienna. Today, I'm here to share our research findings recently published in the PLOS Computational Biology Journal. Our study focused on improving how we detect and classify mouse ultrasonic vocalizations or USVs. I worked on this project with Dr. Peter Balash from the Acoustic Research Institute, Dr. Dustin Penn, Dr. Salazala, Maria Adelaide Marconi, and Doris Nikolakis from the University of Veterinary Medicine in Vienna. To set the stage for my talk, despite your expertise in mouse USVs, let's begin by defining what USVs are. As is the case with human speech, mice use vocalizations to communicate with each other and to respond to various stimuli. These vocalizations are emitted in a wide range from about 3 kilohertz to more than 120 kilohertz. And those beyond the range of human hearing are named as ultrasonic vocalizations or USVs. In the field of neuroscience and behavioral biology, USVs are used as a base model for understanding how mice or even other mammals communicate with each other through sounds. Additionally, biomedical scientists record USVs of genetically modified mice. Then they analyze the patterns of USVs to understand the genetic basis of autism and speech disorders, and hopefully to find potential therapies. To fully leverage the information contained in USVs, it's necessary to analyze them by computing their spectrograms. Mouse USVs are discrete units or syllables on spectrograms with gaps of silence between them. And then experts classify USVs into different classes based on their spectrotemporal features. Although there is no consensus on the appropriate way to classify USVs, according to expert knowledge, in this study, we labeled USVs into 11 classes as shown on the right, with their full names provided at the bottom. To simplify analysis, automated methods are developed for the detection and classification of USVs on computed spectrograms. Several software tools are available for detecting USVs, such as Muppet, DeepSqueak, or DisQ, USVsec, Ultravox, MSA, and AMOD. It's important to be aware that these tools may yield two types of errors: false negatives when actual USVs are not detected, and false positives when noise is detected as USVs. These errors are more likely to happen when working with new data, as the tools were originally developed for a specific mouse strain, sexes, or context. There have been relatively few studies that have compared USV detection tools. For example, Binder and colleagues conducted one of these studies evaluating Avisoft and MSA based on the number of USVs they could detect. In another study, Kofi and colleagues compared Muppet, Ultravox, and DeepSqueak using a small set of ground truths. The second research gap is that USV detection tool performance is not fully investigated using new data sets that differ from the training data set. To our knowledge, one study compared Avisoft and MSA for different mouse strains, and another study has compared MSA and Muppet for different recording conditions. Consequently, it's unclear how well detection methods perform when applied to new recordings that differ from the model development dataset. Once we have detected USVs, the next step is classifying them using an efficient method. However, limited research is available on the classification of USVs, and most existing methods share similar architectures. For example, one of the state-of-the-art classifiers, DeepSqueak, utilizes convolutional neural networks, or CNN, and a data set of 56,000 spectrogram images to classify USVs into 5 classes. However, the generalizability of USV classifiers has not been investigated before our study. This research gap leaves us uncertain about how well these tools can classify USVs for new data set. There are 3 key factors that can make USV classifiers less generalizable. First, they often use high-quality recordings during development, so they struggle with real-world recordings that have more noise. Second, most methods rely on a single model, which may fail to find the most efficient model for classification, unlike ensemble models. And lastly, they use the same kind of data for both developing the model and evaluating it. This hides the model inefficiencies when dealing with different mice strains or different recording conditions. Considering these difficulties, our study aims to evaluate current USV detection and classification methods and develop an improved method that mitigates false positives during detection and also enhances the generalizability of USV classifier. This study uses 2 data sets, a development data set, or DEV, to train, validate, and test the developed model, and evaluation data set, or EF, to assess the generalizability of the developed model. The development data set includes recordings of 41 wild male mice in presence of unfamiliar female. The evaluation data set consists of 2 sub-data sets, EV wild, which includes recordings from 3 wild male mice in presence of female urine, and EV lab, which includes recordings of 2 lab male mice in presence of frozen female urine. It's worth noting that both high and low SNR recordings are included in both development and evaluation data sets. This slide presents the schematic of our developed model called Bootstrap. The audio signals pass through a selected detection tool, then detected elements are transformed into spectrograms, smoothed, and used as input for the classifier. Because the development data set is significantly imbalanced, the inputs are bootstrapped 10 times to increase the generalizability and reliability of the classifier. Then 10 bootstrap inputs are fed into 10 CNN classifiers, trained using a snapshot ensemble learning with a cosine annealing learning rate. The figure below compares the cosine annealing learning rate and the standard learning rate. The cosine annealing learning rate has shown to be more effective because it allows the model to escape local minima by retraining, by using a larger learning rate after initial convergence. In contrast, a standard learning rate leads to convergence at a local minimum at the end of the training. And finally, our classifier outputs 11 classes of USVs and 1 class of noise, which is the average of 10 models based on bootstrap data. We evaluated the performance of 4 widely used USV detection tools, including AMOD, which is developed using wild mice, and Moped USV-Sec and Deep Squeak or DSQ, all 3 developed using lab mice. To optimize the tools, we conducted parameters tuning for all of them except AMOD, which we maintained with default parameters based on discussions with the developer. And we evaluated all tools using 2 recordings from wild mice and 2 recordings from lab mice. Performance analysis was based on true positive rate or TPR and false discovery rate or FDR, with TPR evaluating the omission of actual USVs and FDR emphasizing false detection of noise as USVs. In the previous slide, I showed you information regarding the comparison of existing USV detection tools. This slide shows how we did comparison between our proposed classifier, Bootstrap, and the state-of-the-art classifier, DSQ. To ensure a fair comparison, we considered both pre-trained and retrained versions of DSQ. For the pre-trained DSQ, we employed the original model weights as published in the DSQ paper to maintain the consistency with the model's documented performance. And for the retrained model, we utilize our development data set to train the classifier again. This approach allowed us to assess how DSQ performs when tailored to our data set. And we evaluated the methods using 2 recordings from lab mice and 3 recordings from wild mice. We use a F1 score as a metric for comparison, and as we know, the F1 score is a single value that combines both precision and recall, and it's suitable for imbalanced data set. The figures below shows the performance of 4 detection tools. AMOD and USV-Sec outperform others in terms of true positive rate for both low and high SNR recordings of wild mice and lab mice. And it should be noted again that AMOD developed for wild mice and USV-Sec developed for lab mice, but still they both can outperform others for both lab mice and wild mice. And in terms of false positives, AMOD showed an advantage over USV-Sec, making it the preferred model for the detection step. However, AMOD still produces a considerable number of false positives, which need to be removed in the classification step. As we all know, we are not entirely sure how mice categorize USVs. Therefore, we evaluated Bootstrap with a varying number of USV classes as defined in prior studies. In table below, you can see the results of 6 different combinations of USV classes. The range of USV classes we studied varied from 2 to 12 types. For instance, in an 11 class scenario, we merge ultra short and short into a new class called short, since the primary difference lies in their duration. Or in a 3 class setup, the classes C2, C3, and harmonics were pulled into jumps and harmonics. USVs without jumps were grouped as no jump, and a separate class was designated for false positives. As expected, classifying all 12 classes yielded the lowest F1 score, while categorizing them into 2 classes resulted in the highest F1 score. This slide presents the performance statistics of Bootstrap and DSQ. To make the predictions from Bootstrap and DSQ comparable, Bootstrap outputs were pulled into 6 classes. As expected, all 3 methods, pre-trained DSQ, retrained DSQ, and Bootstrap perform better for the type of mice that were used to train them. However, still Bootstrap outperformed both versions of DSQ for both types of mice. And we see that all models perform poorly in classes U inverse and C or complex. We believe that this can be due to significant difference between the lab mice and wild mice in these classes. And this difference is illustrated by the TSNE plot. In this plot, the green dots representing data from lab mice are located far away from the red and blue dots representing data from wild mice, which is the case in the class UI. On the other hand, for the class RISE, which is well classified by all models, the distance is minimal. With that, I would like to close my presentation by summarizing the main points. We compared the state-of-the-art methods of USV classification and detection based on different recording conditions and different strains. Default A mode is more generalizable than existing tuned USV detection tools, and we developed a classifier, Bootstrap, that outperformed the state-of-the-art classifier due to applying ensembling techniques. Thank you.
Thank you very much for your fantastic lecture. Are there any questions? Yes, Alicia, please come.
Thank you for your nice talk. I have a question because you have shown examples, just one example of harmonic calls, but without vertical scale. Do you remember what was the fundamental frequency of the harmonic calls, approximately?
I think that's around, around 50, I'm not sure. Yeah.
And what was the sampling rate?
Sorry?
And what was the sampling rate of your...
Sampling rate 25 and 255, 250 kilohertz.
Okay, because I was just wondering if maybe some calls didn't have harmonics just because they were at the border of the available range, and maybe the analysis window maybe was too long for quickly changing signal. Okay, thank you very much.
Welcome.
Thank you. Interesting talk. Several things. First of all, when you're deciding whether a call is a real call or not, presumably, what's the ground truth here? Do you have devocalized mice? Do you know what a call really looks like, as opposed to noise?
What do you mean?
Well, in the spectrogram, you showed a false positive, I think, and a false negative.
Yes.
But how do you know which is a real call?
Okay. You mean compared to false positives, or?
Yeah, when you look at a spectrogram,
Okay.
how do you know which are the real calls and which are the noise?
Okay, the false positives are some vertical noises.
Okay.
And
yes.
The real calls are just, like tones. So.
Okay, and here's just a comment. We've played around with vocal map with rat calls, and it's very tempting to collapse call categories
Mhm.
because improve accuracy. But in our case, in terms of rat calls, I think your short and ultra short calls would probably be the equivalent with rats, if there is an equivalent, would be flat and short calls. And we know from drug studies that these calls are distinguishable. You know, some drugs affect one call and other drugs affect another call subtype. So although it's tempting to collapse calls, I would caution about that, depending on the scientific question, obviously. Thank you.
Okay.
Yeah, thanks for your very nice talk. I was just curious about the runtime of the different algorithms. Could you just compare them a little bit? I mean, in particular, your new algorithm, Bootsnap, and
For clustering or for classification or for detection?
detection.
For detection, I think USVsec was the fastest. And then, yeah, we have, I mean, because, I mean, if you would like to have high accuracies, I mean, for example, for DeepSqueak, you would need to have some manual annotations. So I think you have to also consider this. But when you have the manual annotations and you train the network, then it would be fast. I think then DeepSqueak would be the fastest. But comparing AMOD and USVsec, AMOD is faster than, I mean, sorry, USVsec is faster than AMOD.
And so they all run faster than real time, I suppose, huh? They classify much faster than it would take, you know,
Yeah, sure. Yeah, sure. Yeah.
Thank you.
Welcome.